Hello class, this is Dr. Anderson and we are starting a section on metabolism. I am producing a series of videos that I will hope will help you prepare for class, and then we'll be doing some active learning exercises in class with my colleague Dr. DeLay Olson. So let's get started. Here are our learning objectives for the first section. First of all, we want to define some terms: metabolism, catabolism, and anabolism. And we want to talk about a kind of reaction called a redox reaction. Here's an example here: NAD plus and NADH. One form is oxidized, which is and that's a lower energy form, and the other form is reduced. That means a higher energy form. So then we're going to talk about some cellular processes. First, we'll start with glycolysis, where it's occurring, and just essentially the basics of glycolysis, and then a little bit about why we would make lactic acid from pyruvic acid. Then we're going to go on and take the products of glycolysis, namely pyruvic acid, put that into the Krebs cycle, also called the TCA cycle. That's occurring inside the mitochondria in the matrix. And then as we pull the carbons off of pyruvate, we're going to make CO2. So that's where CO2, carbon dioxide, a waste product of metabolism, comes in. Then also we're going to harvest from energy from the Krebs cycle. Those are stored in some high energy compounds, and then those are taken to the electron transport chain, which I often abbreviate ETC. And so in oxidative phosphorylation, as the high energy electrons from those high energy compounds, like FADH2, for example, donates its high energy electrons, and that's going to shuttle through the electron transport chain, and hopefully you know that those electrons are finally picked up by oxygen. So oxygen is the final electron acceptor, and that of course occurs into the mitochondrial cristae. So hopefully by the end of this presentation you'll be able to answer all of these learning objectives. Then we have a second set of learning objectives about metabolic states. We're going to talk about glycogenesis, the synthesis of glycogen, glycogenolysis, which is the breakdown of glycogen, and gluconeogenesis, which is the synthesis of glucose, new glucose, from non-carbohydrate sources. And we'll talk about where these occur. What makes up a triglyceride? We're going to have a glycerol backbone and 3 free fatty acids. And really, we can get more energy in a triglyceride than we can in glycogen, which is why most of our stores are in fat. We'll talk about how fats are transported in the body, how we get fat into adipose tissue with the help of this lovely enzyme, endotheliolipoprotein lipase. And we're going to talk about absorptive and post-absorptive states and why that would be active in a certain way. We're going to talk about what occurs with deamination, what happens to the products of deamination, and finally we're going to finish up with what kind of substrates are considered to be essential. What in the world does that mean? So we're going to start with some definitions, talk about cellular processes, and then talk about handling organic substrates. Those are the 3 major themes of this presentation. Alrighty, so metabolism is defined as the sum of all chemical reactions. Every time you break a bond, every time you make a bond, every time you pump an ion, every time you phosphorylate something, anytime you make a protein, all of those reactions, you add them all up and you get metabolism. And we can divide metabolism into a couple of main types. We can talk about anabolism. Anabolism is storing energy in bonds. So when you take amino acids and you make it into a protein, that's an anabolic kind of metabolism. Catabolism is breaking bonds and releasing that energy. So if you take a glycogen molecule, and you break down the glycogen and release the glucose, send the glucose to the cells for glucose uptake and utilization. Those would be catabolic reactions. Now, many of the reactions in the body are coupled. So as one molecule is losing energy, and essentially what that means is that that molecule is losing an electron or losing electrons, that molecule is oxidized. And so you think about burning. When you burn something, you oxidize it. The thing about metabolism, though, is we're really doing a slow burn. Certainly you can release the energy from food if you put it in a bomb calorimeter and burn it up and release all the heat, but then you don't get to do anything with it. The oxidation that your body does are done in many, many steps so that you can actually capture some of the energy and do something with it. So this term makes no sense, but when we talk about a molecule gaining energy, we say that molecule is reduced. So the oxidized molecule donates an electron, the electron goes to another molecule that gets reduced, and so the gaining of the energy comes in the form of those electrons. So here's an example of a redox reaction. So here's glucose. It goes through several glycolysis reactions, and now we have two pyruvates. So glucose is a 6-carbon molecule. Notice we have 1, 2, 3 carbons in pyruvate. And pyruvate's a lovely molecule. We can put it into the Krebs cycle and make ATP out of it. But the thing is, if we're running low on oxygen, if you remember from our pulmonary unit, we have to have a PO2 of at least 3 millimeters of mercury in a mitochondria in order to drive aerobic metabolism. So under anaerobic or hypoxic states, we are relying more on glycolysis. And glycolysis requires a molecule called NAD+. Energy then is donated to make NAD. Well, if all of your NAD is in the form of NADH, glycolysis can't keep going. So we have a little trick that we do. We take a high energy electron, and then the hydrogens follow to maintain electron neutrality. That electron with the hydrogens come along and get added to pyruvate, and that makes pyruvate into lactate. So you may think of making lactate as a bad thing. Actually, in the short term, it's a beautiful thing because it regenerates NAD+, so that glycolysis can keep going. So here are redox reactions, or reduction and oxidation, happening at the same time. So pyruvate is a low energy molecule. It's in the oxidized form. NADH is a high energy molecule in the reduced form. NADH can donate its high energy electrons, and the hydrogens come along to maintain electron neutrality. And now pyruvate is taken from an oxidized form into a reduced form, whereas NADH is the reduced form and it turns back into NAD+. It's the oxidized form. So this is the most important pair of redox reactions that you should know. I'm not holding you responsible for all the reactions in glycolysis and the Krebs cycle. I just want you to know the main idea. But this is pretty important because under hypoxic situations, we're going to make more lactate. It seems like a bad thing for someone to be in lactic acidosis. Of course that would be. But in the short term, making lactate allows us to keep glycolysis going. And that is a good thing. All right, so we need to remember a couple of things. Second law of thermodynamics says that with each energy transfer, each redox reactions, every time we make a protein, every time we break down an ATP, anytime we have metabolism, we're either storing energy in a bond or releasing energy in the bond, but not all the energy is captured. Some of it is lost as heat. So the thing is, if we measure the heat, and heat is measured in the kilocalorie, kilocalorie is how much heat it requires to take a liter of water from 15.5 to 16.5 degrees centigrade. What a definition that is. Anyway, a kilocalorie is a heat to raise a liter of water 1 degree. So if we measure kilocalories, then that's a direct measure of metabolism. All right. So here are a lot of big things going on in metabolism. We maybe eat some carbohydrates. Those are broken down in the gut. You reabsorb the sugars, or you absorb the sugars. The sugars would go into a cell. In the cytosol, we'd find the enzymes for glycolysis. We would take a glucose molecule and break it in half, and then we've got a pyruvate. If we've got oxygen, we can take that pyruvate and put it in the mitochondria and burn it in the Krebs cycle. If we don't have oxygen, we can reduce pyruvate and make lactate. That lactate then regenerates NAD+, so glycolysis can keep going. However, pyruvate is not the only thing that can go into the Krebs cycle. We can break down fats and proteins. We could deaminate an amino acid, and then we have some carbons we could put into the Krebs cycle. We could take a long-chain fatty acid and break it down into groups of 2. That's called beta oxidation. We could put those byproducts into the Krebs cycle. Notice as we're pulling off the carbons, the broken off carbons become CO2, and the energy in the bond is used to make some high energy compounds. So there's some ATP that's made directly. We also have some high energy compounds that go into the into oxidative phosphorylation. Oxidative phosphorylation takes high energy compounds and turns it into ATP. Notice that process takes oxygen, and so oxygen accepts the high energy electrons. Hydrogens come along to maintain electroneutrality, and the byproduct then is water. So that's the big picture of metabolism. Here's another overview diagram that I like a lot from the textbook Vander. So glycolysis, you take a glucose, you break it in half, you net 2 ATP and some NADH. NADH can go into the electron transport chain. Pyruvate can go into the mitochondria. So this is happening in the cytosol. This is happening in the matrix, or the fluid part of the mitochondria. And as we take pyruvate and we send it through this enzyme pathway, we are pulling off a carbon from each pyruvate. So we've got 2 CO2s, and now we have 2 acetyl-CoAs that go into the little circle here that scares the bejesus out of students everywhere. And we are pulling off carbons and producing CO2, producing a little bit of ATP, but also producing some high energy compounds that can then go to the electron transport chain. And as those electrons shuttle through the cytochromes, we create a hydrogen gradient. That hydrogen gradient then can be used to basically drive ATP synthase, and we make a bunch of ATP. O2 is the final electron acceptor, and those then get converted into waters. So you see there's a mole of glucose, 6 moles of oxygen, ADP, and some phosphates, and then we get the carbons in glucose become CO2, the oxygen becomes water, and we make a bunch of ATP. So glycolysis starts with glucose. There's a couple of ways to display glucose. Here we kind of see the line diagram and then the bonds to show you that it's really kind of a circle. So glucose is a hexose. It's got 6 carbons. There's There are a couple of other hexoses. Fructose is a hexose, galactose, glucose and fructose make a sucrose, that's table sugar. Glucose and glucose make maltose. Glucose and galactose make lactose, you know, all kinds of wonderful things. And all of the hexoses can be put into glycolysis at some point. As I said previously, what you're doing is taking a 6-carbon molecule, breaking it in half, and making 2 3-carbon molecules. So glucose is a pretty stable molecule. So we actually phosphorylate it a couple of times to destabilize it, and then we net make 4 ATP as a result. So what we net are 2 ATP. The energy in the bonds make NADH. This goes to the electron transport chain to make ATP. This process is fast and does not require O2. So then we can take pyruvic acid, take off a carbon, make another NADH, and now we've got a 2-carbon molecule called acetyl-CoA, which goes in the Krebs cycle. Acetyl-CoA can be used to make fat. You can string carbons together and make an even carbon free fatty acid, but we can't go back to pyruvate. This step is not reversible, and this will come in handy later when we start talking about a process called gluconeogenesis. That means acetyl-CoA is not gluconeogenic. So we put pyruvate into the mitochondria. We make acetyl coenzyme A. We join it with a bunch of intermediates. And as we're pulling off the carbons, we are storing energy in ATP, and then we have some high energy compounds, NADH and FADH2. These are going to go over to the Christie, where they're going to find the oxidative phosphorylation. The carbon from the broken bonds is given off as CO2. So in oxidative phosphorylation, there are high energy compounds, and we have a bunch of cytochromes in the Christie of the mitochondria. And essentially, as we donate this high energy electron, the electron shuttles through the cytochromes, and the hydrogen then goes into the little membrane, or the little space in between the membranes. Remember, the mitochondria have 2 membranes. So as hydrogen builds up in there, you've got a gradient, and hydrogen can flow across that gradient, and that's what allows us to make ATP. So we don't have electrons jamming up in the cytochromes. We have a way for those electrons to finally jump off. They jump onto oxygen, and hydrogens come along, and now we have these wonderful water molecules. So glucose gets converted to CO2, oxygen gets converted to water. And here we are back at our same overview: glycolysis, the Krebs cycle, our high energy compounds goes into oxidative phosphorylation. So we think about, in the past you may have thought about the Krebs cycle and oxidative phosphorylation as sort of separate things, and they're really not. They're on a continuum. If you have hypoxia and you can't accept these electrons, the electrons jam up, the high energy compounds have nowhere to go. This stops, everything stops, because they're very intimately connected. Now, glucose can be stored. Glucose can be stored as glycogen. Remember, there are lots of carbohydrates. Cellulose would be in plants, and we can't do just that. Starch is in plants. We can use that as a food source. And animals store glucose as glycogen. And you have big, long chains of glucose to form glycogen. There are glycogen stores in the liver, and we can use those to raise blood glucose level. And there are glycogen stores in the muscle, and we can use that to serve the energy needs of the exercising muscle. So you can take glucose, put it into a cell, it gets phosphorylated, and most tissues of the body, we can break down glucose into pyruvate. You can take pyruvate and turn it back into glucose, and we'll talk about that at another time. The liver is able to take this phosphate off the glucose, so the liver then can raise blood glucose level. The thing is, if this were muscle is not able to take off that 6-phosphate. So muscle can break down its glycogen and then put the phosphorylated glucose into glycolysis. But the muscle cannot break down glycogen and then put the glucose into the blood. So it's the liver that does that. Now, there's some data that kidneys can do some things in terms of glucose metabolism, and we're just not going to cover that in this course. We're going to focus mainly on the liver. All right, gluconeogenesis. Gluco is sugar, neo is new, and genesis is creation. So we're making new glucose out of non-carbohydrate sources. So here we have lactate, 3 carbons, pyruvate, 3 carbons, some amino acids, all of which are 3 carbons, and glycerol, 3 carbons. The liver can take these 3 carbon molecules, put them together into 6 carbon molecules, and make new glucose. We would want to do that during the fasting state or the post-absorptive state. You wouldn't want to do that when there's plenty of glucose around. So here's our glucose. We can burn it in glycolysis. We can make a pyruvate. We can take a pyruvate and turn it into glycerol and make triglycerides. We can take our pyruvate and make it into lactate. We could take amino acids and deaminate them and make them into pyruvate. Those could then be burned in the Krebs cycle. Amino acids can go into the Krebs cycle. Just all kinds of crazy things can happen. But if we have a pyruvate or amino acids or lactate or glycerol, we can all convert them into precursors that could take us back to glucose. Glucose 6-phosphate in muscle, or and then the liver can take that phosphate off, and now we've got glucose to raise blood glucose level. So lactate, some amino acids, pyruvate, glycerol can all get taken back into glucose. But looky here, here's acetyl-CoA. This arrow doesn't go back this way. This is only one way. So once we're acetyl coenzyme A, we can certainly burn this in the Krebs cycle. We can turn this into fat, but this is never going to be turned into glucose. All right, here's some triglycerides. So we have a glycerol backbone. We bind those covalently to free fatty acids. So there's a triglyceride. There's a phospholipid with there's the polar head and the 2 tails. Now notice we have a couple of kinds of fatty acids. We have one that's saturated. Saturated means there's no double bonds. Carbon can bind to 4 things. If it's bound to 4 things, then the carbon is saturated. If we have some double bonds here, see, this carbon is only bound to 3 things. 1 carbon, a hydrogen, and then a double bond between another carbon. So these are unsaturated fatty acids. Now what's neat about these, it's harder to make cholesterol out of these. These tend to be plant fats, and they tend to be healthier for you. Saturated fats is what we tend to find in animal fats, which is why, so we have nice straight lines and they all pack in together, so they're solids at room temperature. These are liquids at room temperature, so You want to use your polyunsaturated fatty acids. Those are better for you because they are less. One reason is they're less likely to contribute to the bad cholesterol. Now, somebody got the great idea. Oh, well, we'll take saturated fats and we'll put double bonds in them. We'll make them trans fats. Well, evidently that doesn't work. You just want to go ahead and have the olive oil or the canola oil or something like that. You want to go ahead and have the vegetables. You want to go ahead and eat your nuts, your pistachios and your almonds and that kind of stuff, because they're better for you. Turns out the trans fats cannot fool Mother Nature. They're not any better for you than the saturated fats. That was a little bit of a tangent, but hopefully useful. All right, so if we want to burn fat, the first thing we have to do is lipolysis. So we have to take our glycerol and our 3 fatty acids. We need to break those bonds. So we've got a free fatty acid, and then the free fatty acid has to go through a process of beta oxidation. So you break off 2 carbons at a time, and as you're breaking off 2 carbons, you've got an acetyl-CoA, which can go into the Krebs cycle and be burned. Then you break off another 2 carbons into the Krebs cycle and be burned. So fatty acids is a wonderful substrate to make ATP out of in the Krebs cycle. I don't know if I want to go into this right now. You can have acetyl-CoAs build up. They can interact and form something called a ketone. Perfectly nice molecule you can use for energy. You also think about ketones building up with diabetic ketoacidosis. We can talk about that a little bit later. Burning fatty acids, you got to break down the triglyceride, take the free fatty acid, it undergoes beta oxidation. The 2 carbon elements are acetyl-CoA that can then go into the Krebs cycle, but you need oxygen to burn fat. Now, how are we carrying our fat around? We'll talk about this at another time, but the ingested fat is packaged into something called a chylomicron, and it goes from the gut into the lymph, from the lymph eventually into the venous blood, and then circulates throughout the body. Your liver can make some lipoproteins, a VLDL, which carries triglyceride around from. So if you eat fat, it's carried around as a chylomicron. If your liver makes fat, it's carried around as a VLDL. To get fat into adipose tissue, you have to break it down with endothelial lipoprotein lipase into a monoglyceride and 2 free fatty acids. And why is that? Because a triglyceride is lipid soluble, but it's too darn big to get through a membrane. So you got to break it down to get it into the adipose cell. As adipose tissue pulls off fat from your VLDL, you get into something called an intermediate density lipoprotein. That will get you back to the liver. An LDL, lipoproteins with a lot of cholesterol. So some similar backbone proteins to VLDLs, but instead of triglyceride, we have a whole bunch of cholesterol. So LDL carries cholesterol to the periphery. It indicates mobilization of fat. And LDL is more likely to contribute to atherosclerosis. HDL tends to pick up cholesterol for excretion in the bile. HDL is much less likely to get oxidized, much less likely to pack fat and cholesterol into the wall of a blood vessel. So we think of HDL as the good cholesterol. If you have great big HDLs, it'll pick up cholesterol and keep your cholesterol low and give you a nice heart health. LDL cholesterol, high levels of that seems to be associated with not as good heart health. Amino acids, remember we've got a central carbon, a carboxyl group, an amino group, a hydrogen, and some kind of remainder group. They can be nonpolar or polar or ionized, or we can organize them into acidic, basic, and neutral. With the amino group on, you can put them together and make them into peptides. But if you want to use an amino acid for energy, you've got to take this amino group off. And that process is called deamination. So here we have an amino acid, and we take off that amino group, and now we've got a keto acid. The liver can use keto acids for energy. Other cells can too. And then we've got an ammonia, and we don't want ammonia running around loose, so your liver takes that ammonia and turns it into a urea, and then your kidneys can help you excrete some of that urea. You also can do something called transamination. I can take an amino group from one amino acid, put it on a keto acid, and now I've got a new amino acid. So, big unifying diagram. Here's protein, which can be broken down into amino acids. Amino acid can be deaminated. The ammonia is used into urea. Amino acids can be turned into pyruvate, used to make glucose, or amino acids can be put into the Krebs cycle to make ATP. Here, glycogen is a gigantic molecule that can be broken down into glucose. Glucose can be burned in glycolysis, turned into pyruvate, can be turned into acetyl coenzyme A, burned in the Krebs cycle to make ATP. Glucose, through glycolysis, can also be made into a glycerol, or glycerol can go into some pathways and be made into pyruvate. So pyruvate, amino acids, glycerol can be turned back into glucose by a process called gluconeogenesis, or glucose can be turned into pyruvate and burned in the Krebs cycle and used to make ATP with oxidative phosphorylation. We can break down fat. Glycerol can be used to make glucose. Glycerol can be burned to make ATP. Fatty acids can only go into coenzyme A and be burned in the Krebs cycle and oxidative phosphorylation to make ATP. So fatty acids are not gluconeogenic. They're actually ketogenic. Glycerol and pyruvate are gluconeogenic. Lactate is gluconeogenic. Amino acids, some amino acids are gluconeogenic. Some amino acids are actually ketogenic. And of course, we're not going to talk about which ones are which, but lots of amino acids can be used to make glucose, but not all of them. Some of them end up making ketones. All right, here we are. So essentially what I wanted to do was give you some background on metabolism. Hopefully you'll have some time to view this presentation over the week so that on Friday when we do some active learning in class, you'll have some exposure to the concepts. Thank you so much for listening, and I'll see you in class, or I'll be talking to you in the next presentation. Bye for now.